Okay, so lab 4 was quantifying proteins. We essentially used the Bradford assay to do that. And remember that Bradford was that blue dye, and when it comes into contact with protein, it lights up blue, like a brilliant, oh wait, actually blue. It lights up blue. No protein or very little protein, nothing. So Bradford assay, we were able to see and measure this blue using the spectrophotometer at nanometer 595. And just in water, same thing at 595, but nothing. So this was our first step towards quantifying proteins. As we saw, proteins are super important, and in this case they're in plant cell. Lots of proteins are inside the cell, they're doing things, they're enzymes, all that. There's a lot of surface proteins too. A lot of the times they're receptors, sometimes they're channels, things like that. So we made hypotheses based on, whether the plant root or the plant leaf would have more or one of the other type soluble or transmembrane proteins. And we did this through kind of just mashing them up in the cell. But I did want to go over Bradford one more time. So yeah, let's get rid of all this. Okay, so when you make a Bradford graph, you always have to label your axes. So I'm going to say concentration. And remember, concentration is substance and volume. So in this case, it was just micrograms, which is the substance, and volume per microliter. So you can just remember that. And then the absorbance values from the spectrophotometer. And these are usually small, like 0.27, something like that. So what we did was have 4 specific concentrations. For our purposes, let's say these were at, so starting lowest over here, this was at, I think it was like, let's say, I don't know. These weren't the actual values. Let's say 5, let's say 10, oops, sorry. And then 20, and then let's say over here it was like 40. It's getting a little heavy over here. Let's just use this as 15 right here. Okay, so we have, let's say we want to measure at these concentrations. If we measured here, and we know that we have a certain solution, so if we make our own solutions and we know, like say we use milk protein as the control, and we have 4 tubes, and in one of them, there's 5 concentration. In the other, there's 10, 15, and 20. So the amount of blue we're gonna see is really heavy in this one, pretty good here, not much here, and barely any in there. Because the more protein you have, the more Bradford reagent's gonna shine blue. So when we do absorbances, so we know we're at 5 concentration because we're measuring this first one. First one's here. Here, higher absorbance. Three, 4, and these were the values that you found. And they correspond to given concentrations because we know the concentration, and right now we're making the standard curve. It's not really a curve, but it helps to visualize it. So now that we have our standard right here, we know we can measure the absorbance and we'll know a concentration. So say we have our unknown protein and it measures at this absorbance. That means that we can go over here and measure it exactly down here at that absorbance. And we're gonna know the exact concentration too that it's gonna correspond to. Somewhere like 17 maybe, like that'd be my guess based on where that's at. Hopefully this was clean enough, sorry about that. Got a little busy, but that should be good for this lab.